Welcome everyone, and for those of you that don't know, SC 2.0 is a project to synthesize a highly modified version of the yeast genome Saccharomyces cerevisiae. We have partners all over the world. There are labs in China, Australia, Singapore, and the UK. And we have representatives, I think, from most of those teams here today. And we've been we've split the project up by chromosome and assigned different chromosomes to different teams. In fact, one of the projects going on in my own lab right now is to synthesize chromosome 4, which is the largest amount of synthesis needed for a single chromosome in yeast. And we are very pleased to be joined on this by our latest partner in this effort, which is Tokyo Institute of Technology in Japan. Yasaisawa, you'll hear from him later today. As well as Jungyao Dai's group at CIAT. And the Joint Genome Institute, which helped us with some of the synthesis. I'm going to start skipping ahead here since I don't have any AV, and just tell you that we're basically somewhere between 95 and 98% done with the synthesis overall and assembly of this genome, and we're in the finishing stage. There we go. And you can see our many partners here, and this we'll show you the design is completed, the 6 chromosomes that have been completed and published on last year, and then the status of the remaining chromosomes, approximate levels of synthesis and assembly. Oops. So I wanted to mention that I mentioned chromosome 4. The largest chromosome. Now I'm going to shift to chromosome 1 and just give you guys a little taste of the science that's going on in the lab that I think shows the power of doing this type of work. We also focused on chromosome 1, which we should have finished a long time ago. It's a tiny chromosome. But it's been one graduate student's project, and she has gotten highly distracted, as you'll see, as a consequence of what happened. So chromosome 1 was a very interesting case because it actually has a huge amount of repeated DNA near the telomeres. And part of our design was to remove all repeated DNA. So we worried that if we downsize the chromosome so much, we might make it much less stable than it should be and create a problem. Also, part of our overall plan is to add a 17th chromosome, a so-called neochromosome, that has all tRNA genes on it. And that would make 17 versus 16 in the native yeast. So we decided somewhat arbitrarily to fuse chromosome 1 to another synthetic chromosome. And so Jingchuan Wo in the lab looked into that, and we decided if we're going to try to see if we can do this, but it wasn't obvious because the 3D structure of the yeast genome first reported by Duan et al., is very non-random. It has a very distinct order to it. And we wondered if moving one chromosome onto the end of another would have an impact. So we decided if we're going to try, let's try fusing it to the shortest arm in the genome, a medium-sized arm in the genome, and the longest arm in the genome, and what would happen if we did that. And to do this, this sort of shows the problem. So here's the 3D structure, kind of hard to see what's going on. Now we've grayed out the chromosomes that are irrelevant, still hard to see what's going on. Here's chromosome 1, way out on the periphery and near this region, which is where all the centromeres are clustered together. And here in the arrows are the positions at which we're going to fuse chromosome 1, thus dragging it completely out of its normal context. What's the consequence of that? So Jingchuan used a CRISPR-based method to perform this chromosome fusion and discovered that it was actually very easy and very efficient. And so at a somewhat magical committee meeting, Brendan Cormack suggested to her, well, why stop now? Why don't you just keep fusing the chromosomes together? And that got us really thinking about why do species have different numbers of chromosomes. So here on this beautiful tree of life in which Earth birth is at this position and the origin of life is about here, and time going outwards, you can see the modern organisms on the outer edge and all the lineages leading to those. And we see dramatic differences in N, the number of chromosomes in different organisms, ranging from the hundreds in the fern species to the world record holder, the jackjumper ant of Australia, which amazingly in 1986 was shown to have just a single chromosome. So in stark contrast to that, a butterfly, which is relatively closely related on this tree, has N of 223. So why is this, and how did this come to be? So we wondered, could we do something to make the yeast into something closer to the jackjumper ant, a kind of jackjumper yeast? And so she came up with a blueprint for how to fuse the chromosomes together. And she worried that the 4 chromosomes, maybe we could get to 4 chromosomes, but they should probably all be roughly equal sized. And that guided her thinking. And then she thought, well, maybe we'll be lucky and we can go to 3 or 2 or perhaps even 1. And to make a long story short, she succeeded rapidly in getting to 9 chromosomes and then to the 4 equal-sized chromosomes and more recently to 2 chromosomes. And I should add that we had made the miraculous discovery recently that another team working in China, blissfully ignorant of our effort and vice versa, basically actually took it all the way down to 1 chromosome. And they have very similar results to ours. The results include that the cells are remarkably healthy despite these massive changes to their genome structure. So this is the 3D structure determined by our colleagues at Pasteur of the native N equals 16 yeast, and this is the chromosome structure of the N equals 3 strain. So you can see the red centromeres are still clustered together, and the telomeres cluster together on the nuclear surface, but other than that, the overall structure has been massively changed, and despite this, the cell is quite happy. Very interesting and provocative results suggesting amazing plasticity of the genome. I want to just end by saying that I'll say a few words about our Center of Excellence in Genome Sciences, or CEGS, and it is aiming at a very different target, which is understanding human genetic disease at a much finer level of detail. There have been many, many hundreds of genome-wide association studies performed that have identified many plausible causative variants in the human genome, but it's very hard to pinpoint exactly which change or changes in each of those regions is responsible for the genetic disease. And this is something we've become very interested in. Most such associations lie in the so-called dark matter of the genome, or the non-coding regions, and the combinations available in nature usually do not allow determination of the functional of the individual genetic elements that might underlie the disease. So what we want to do is to isolate each of these elements in its native context to identify the important causal alleles. So we've reported previously on our ability to build big DNAs, mammalian DNAs of 100 kb and up, that we can use to dissect these GWAS hits by generating and testing synthetic haplotypes in stem cells and in engineered mice. And we're very happy to have recently reviewed news that our proposal to the National Human Genome Research Institute, which aims to develop this technology, will be funded, and the details of that are at the bottom of the slide. I want to emphasize this important collaboration with Matt Morano, Aravinda Chakravarty, and others. So with that, I will stop and hand the podium over to George.
Thank you very much, Jeff and Nancy. Sure. And my short time here will be to bridge this introductory comments with the next panel, which is on announcing this new international project and how we're going to coordinate it. And I just want to get down to the nitty-gritty and set that up. This is my conflict of interest slide, but I have a cleaner one, which is just the ones that are for today. And the slides are online, and you probably have figured out the Wi-Fi by now. We have a huge growth in cumulative funding here. This is sort of centered around a 5-year window around today. I call it Genome Project, or we call it Genome Project Write and Test, because it's important to do testing. We're doing radical enough writing that we have to test it, as Jeff nicely showed with 3D structure and function. And anybody who wants to contribute to any of these documents that I'm showing here, please feel free. I'm sure this one's got some problems. Our consortium now has 106 institutions, at least in 15 countries, and we encourage more to join. This is not meant to be an exclusive group, and this is not a secret meeting, by the way. Neither was the last two. The reason that we're even contemplating this is because of this extraordinary exponential curve. In fact, it's super exponential because we have factors of 10 on the y-axis, and it's still going hockey stick, not just for reading DNA, which has been a 10 million-fold improvement over 14 years, but also for writing of oligonucleotides, not genomes, not even genes. But they're both sort of in that 10 million-fold improvement range, and we want to get better at both. We have some changes in, there's no way I'm doing this in 2 minutes, I'm sorry. I got 10. So this is, we have the science paper. 2 years ago we had a meeting here, and this science paper was under embargo. We have a few new things that we've been talking about a little bit, but these are formally part of the new project. The emphasis is on that one in the green box, which is virus resistance, recoding we want to be able to develop technology to recode microorganisms, plants, animals, and human cells. Human-induced pluripotent stem cells is quite attractive because we can do rapid testing of the epigenomics, and we'll come back to that. I'm not going to go through this right at the moment. We'll leave it up. That slide will be up. All these slides are available online, but that one will be up at the end when we transition to the panel. We have a choice of literally writing every single base pair or writing the base pairs that we change or something in between. I call this precise hyper-editing. This is not editing in terms of bashing. This is actually getting what you want. It's related to MAGE, or multiplex automated genome engineering, which we use to engineer 321 pieces of DNA to get a genome which we tested, so the test part of GP Write and Test, for non-standard amino acids, biocontainment, multivirus resistant. It was resistant to at least 4 out of 6 viruses, thanks to some work from Baron Isig's group and these authors down below. Moving to human genome recoding, this is the most recent codon chart that I know of, and I think we're settling on, or my group at least is settling on UUR, UUARG. There's 230,000 changes to make, so that's not 6 billion, but it is quite a few changes. And this might be done either by synthesis of large blocks or by hyper-editing. And we can go through this slide in more detail if you like, but there were essentially 3 amino acids that have 6 codons, and this one is a particularly nice one. Now, it's not just about exons and protein coding regions. There are other things that we want to engineer. So, for example, for the community effort, we might want to engineer centromeres or something else so we can swap chromosomes out. Might want to stabilize our creation and all these various repeats. So my lab and others are interested in repeats, and they are not only hard to engineer, they're even hard to sequence. Some of these things are still not sequenced after my colleagues declaring victory on the human genome many times. It is still, the centromeres in particular, are not done, but people are already engineering them. And I love this paper in 2017 where you can get rid of the Y chromosome. The world would be a better place. Anyway, and you could also, and there are various different methods we can use, and we'll come to that in a moment. We've got one of the, I'm just showing this about transplants to indicate that we got rid of 62 endogenous retrovirals, which is a repeat, simultaneously in living pig cells and living pigs. We had to, this is toxic, so double-strand breaks are toxic, whether they're talons or CRISPR. And so we used 2 tricks for detoxifying that. And you can see in the red down below, in particular, the number of clones that survived cloning were essentially 0 on the left and close to 100% after the 2 factors that we used. So that's one way to do it. But that's only 25 double-strand breaks that made it into living pigs. The other possibility is deaminases, and this is something I have to credit Luhan Yang, both with the previous work and with this one. And she started this publishing on this in 2009 using the tau, and we have an announcement from Selectus on tau, and they're great because they're hyper-specific, but in this case they also don't make the triple helices, and we find that these are very toxic, not just double-strand breaks, but single-strand nicks and even editing. So anyway, we're working on new deaminase editors based on tau. Both C to T and A to G, and there's been very excellent work from David Liu's lab at the bottom 2 articles there. Corey Smith has used some of David's lab's base editors to look at the line elements. This is a highly repetitive family, about 20,000 of line 1s, and you can see that we can get cells after a short period of time which have about 50+% of them modified. But then even this mild deamination editing results in lethality. And the most we've gotten so far, tentatively, is at day 11, we've got maybe 400 edits. It's still a record for edits in mammalian cell, but we need to get better at it. And we think that the tau without nicking is going to do it. So the second and final topic is this radiation-resistant, senescence-resistant, immune, so forth, enhancement. Here there are universal cells that you can transfer from one human to another in order to fight cancer. These are universal CAR T cells. And these can be done by zinc fingers, talons, and CRISPR, and they're in clinical trials. But the thing that's exciting about both the organs and the universal donation of organs, even as far as xenotransplantation, is the possibility of enhancement to resistance to pathogens, cancer, senescence, radiation, and cryopreservation. And there's some precedents for this. The little tardigrade down there is one of the animals that can live in the vacuum of space, being heavily irradiated and bounce right back. And so we're moving genes. We have already moved some genes from it into human cells. Organoids, we want to do disease modeling, variants of unknown significance, and so on. We have a new transcription factor library. This is a resource for the entire community, GP Write and beyond. This will be distributed by Adgene and other nonprofits, and we will be submitting for publication any week now. And we can use this not only to make any tissue type, every tissue type we've tried to make, we've succeeded about 15 so far, but these organoids are finally vascularized. So you can get these complex brain-like organoids that Magdalena Lancaster and others developed in 2013, but now they're vascularized, and that allows them to get bigger than a 0.5 millimeter. This is work in collaboration with Mark Scott and Jennifer Lewis at the Wyss. And we've made oligodendrocytes that can wrap axons with myelin. All those cells that we use are from the Personal Genome Project rather than Henrietta Lacks, which is a much abused person and families, and I urge you to read this book if you haven't already. Instead, we do the opposite, and this is the world's open access. This is your resource as well. If you want it, the NIST and the FDA are using our cells, as is the NIH ENCODE project. So this is truly open, well consented, and in multiple countries down there at the bottom. And this is my last couple of slides, is we have this, you can go to this website and you can see some of the papers that have been published on this, including the first CRISPR paper on using a single guide RNA in humans, using guide RNA libraries in human stem cells, and a first comparison of talons and guide RNAs to exactly the same base pair there in red and green. And we also, all these different cell types are listed. And I just want to thank, before I go on to the slide that will transition to the panel, I'll ask Farron and Pam and Jeff to come on up. Farron and Pam are there acknowledged on this slide along with our segs. So Jeff mentioned that he has a new segs. We have an old, three old segs that led to where we are today. The previous one was helped us develop the zinc finger, talon, and CRISPR methods, and the new one is for the testing part in organs and organoids. So I just leave this one up here. Might stimulate some discussion, or we can refer to it. Thank you very much.
I just want to show a couple more slides that are relevant to the panel, perhaps, to introduce the idea of this recoding project and explain where it came from. So the Scientific Executive Committee has been meeting roughly monthly, and we've been debating, what could be a great community project to pull GP Write together. And after a lot of discussion, we decided the idea of the ultra-safe cell line with the emphasis on recoding was really the best idea out there, in that it's genome-wide, required non-trivial amounts of changes to the genome, and perhaps most importantly, could have a big impact, because recoding for virus resistance in particular would transcend species boundaries. So George just reminded me, oops. George just reminded me to show my disclosures. And so the community-wide project has this potential to span many species, and in short, it involves deleting one or more codons from the genetic code table, and then deleting the corresponding machinery to decode that. All viruses would depend on that corresponding machinery and therefore be unable to replicate. And work from Farron and George has shown that indeed this works in E. coli for a stop codon, and moreover, this leads to decreased susceptibility. So there's really strong proof of principle that if this can be done in a human cell line, not just recoding, but actually deleting the underlying decoding machinery, then there's a very strong first principles argument that it should work in mammalian cells. So how might we deploy a project like this to the larger GP Write community, I think, is the main theme we want to talk about in the panel. The basic strategy could be very similar to what was done in SC 2.0, using relatively short DNA segments, perhaps in conjunction with a hyper-editing strategy, to replace the exons of genes, which, of course, in mammals are very short. The target cell would be some sort of stem cell. In human case, would probably be an iPS stem cell. Important questions that are not yet answered, and we hope will be part of this discussion, are what should the target cell line be? So for mouse, it would probably be C57 black 6 embryonic stem cells, but what cell line should best be chosen as a base human cell line? And I think George will probably argue for the Personal Genome Project that you just heard about. Those cell lines, there are a lot of reasons why they would be a great choice. That cell line, once chosen, would have to be distributed to all participants. All the participants would have to agree to some common set of rules, and we did lay the groundwork for this in the SC 2.0. For example, centralized and systematic design scheme, And one very important question, technical question that needs to be discussed is the end game. If we have different teams synthesizing different chromosomes around the world, who's going to put Humpty Dumpty back together again and combine all those chromosomes into one cell? And finally, I think we should add on to this what we call the mammalian genome scramble, where as in the SC 2.0 project, we added LOX-P sites systematically throughout the chromosomes. And by flipping a chemical switch, we can turn that system on and create many variant genomes from one starting genome. And we actually have a set of papers in press on this, Nature Communications. This should be coming out in the next month or so, showing how this can be used to improve the traits of the yeast in a broad variety of ways. So what I would propose is, since a typical mammalian gene has tiny exons and gigantic introns, we would insert the LOX-P sites 10 base pairs in from the 5' end of every intron, and this would allow so-called scrambled exons, which is different from what we've done in the yeast. We haven't put them with every intron, we've just put them with every gene. So this is, I think, something that could be added at little to no cost, because this would be right next to each exon, which would be recoded. So with that, maybe we can begin the discussion.
So I'll just start by asking, so that those would be in tandem repeats, essentially, or they can be either duplicate or invert, right?
Right, George is referring to the fact that the LOX-P sites that we use are symmetrical, fully symmetrical, which means that they can recombine in either orientation equally well.
And second question, have you figured out, so that was presumably some dye like lycopene or something that you optimized. Have you figured out why scrambling the genome resulted in higher production of that compound?
So in the example that was shown, that actually involved scrambling the pathway rather than the genome. But we've also done one of the papers, actually several of the papers, show specific scrambled regions that are responsible for phenotypes, not necessarily production of carotene, but other phenotypes.
And I think, can you remind me, Jeff, what's the status of the introns in your yeast synthetic genome?
They're gone. They're toast, baby.
So they're all completely gone?
Well, they will be eventually.
So you think you can delete all of them?
We've deleted the vast majority of them in the individual chromosomes. Of course, we haven't combined. We have an end game too on the yeast genome, which we haven't done yet. But there is one set of introns that we have not deleted, which is the introns in the ribosomal protein genes, because there's some evidence that deleting those gives rise to a fitness defect. But as teams complete their chromosomes, we ask them to systematically delete those last introns, and then, as needed, adjust the levels of expression of the ribosomal proteins. So we're pretty confident we're going to be able to delete all of the introns. There's one other intron, very interesting, that we're not deleting, which is the HAC1 intron, which is spliced out by a different machinery and is very important for stress response. We have some questions.
There's a question.
In the audience.
Yeah.
Right here.
There's one.
Question.
A question. In the sketch that you laid out, there wasn't a clear role for those of us in the consortium who are from the software and design perspective. And I'm wondering if that's something that would be open to discussion. I noticed that the degree of centralization is more than some of what we work with in more of the design-focused communities, and there are some possibilities for bringing in tooling to support some of the things that right now may be a burden on that central point and a bottleneck.
I think software is going to be absolutely crucial. It may be the most crucial technology we develop. This is a technology development project. We're going to need it for not just design up front, but also analysis and feedback and understanding the testing part. So what do the chromosomes look like, for example? That's a big challenge for mammalian. I mean, Jeff made it look really easy for yeast.
It's because I didn't do it.
Yeah. Anyway, it's never been done for mammalian genome, but it will be soon. But we need the software for really making sure we rigorously test that the epigenomics is properly captured, that it can differentiate in every cell type. So yeah, it's huge. Yeah.
So to your point, though, I mean, I think there's going to be two kinds of software development and involvement in the project. There's going to be community-wide efforts that will benefit not just this particular community project, but I think there also has to be, and I'm speaking now as head of the SC 2.0, where we made this decision very explicitly, there has to be one group that's responsible for the design, and then the rest of the groups have to be willing to accept that design. They can be part of the decision-making leading to, okay, what are we going to do? Are we going to include LOXP sites after every intron? Is that a good idea? Or are you out of your mind? You know, and then, but once those decisions are laid down, then there has to be an implementation, and one group needs to be responsible for rewriting that genome sequence and then delivering that specification to each team. So do we want help with that? Absolutely. And exactly who's going to be doing that, we haven't decided yet.
I might call some of those assumptions into question.
Okay.
So I think that, you know, it's entirely reasonable that the architectural designs, like, you know, somebody's got to figure out, do we want LOXP sites or not? Absolutely. There needs to be some group that determines those architectural designs. But the implementation of them into the genome, you need to have some group who's in charge of certifying that those architectural designs are being complied with. But with, you know, this isn't necessarily even a matter for new tooling. There's existing tooling out there that's been developed in other communities that might be imported to bring in, you know, versioning and, you know, merges and things like that that may be able to help.
Totally agree with you. And I should have mentioned one of the other things that I think was really great about SC 2.0 is we delivered the spec to the teams and we said, here's one way to build it. You build it any way you want, you innovate all you want, but you need to deliver to us the specification. And so what we did, to speak to your point, is we had each team, you know, sequence their chromosome, and then we had another team verify independently the sequence that was obtained. And that is actually, as you point out, it's a non-trivial problem, even for yeast, where it's much easier than it will be for human.
It was a good slide, merging your chromosomes into a lower number. But what happens on the other side if you split existing chromosomes into, if you increase the number of chromosomes?
So we haven't done a whole lot of that. There is another group in Japan that did quite a bit of that, and they were able to make very large numbers of chromosomes. But I think one of the questions that arose about that work was whether whether the number, the copy number of each chromosome was truly maintained, or whether it started to fluctuate, because there is good evidence that short linear chromosomes show enhanced instability, both in terms of increasing and decreasing copy numbers. So that would probably be the major limitation.
Just keep it close to your mouth.
Hello.
Keep
So you
talking.
mentioned that you've mentioned that you've been meeting monthly and discussing this, what you've now announced as the first community project. Do you mind talking through the decision-making process, what other projects were considered that might be sort of follow-up projects, and then maybe what are the, now that this community project has been announced, does that sort of put everything on the back burner, or are there other projects that you could imagine going on simultaneously?
Okay, so well, welcome anybody else on the panel's input into this, but so one thing I think we want to make clear is that we're not in any way, shape, or form backing off from technology development as the main focus of the project. But I think a lot of people really wanted to get involved in a big project, a specific project. And so that's what we tried to steer the committee towards. And the committee meetings were very interesting, and I think the transcripts of the committee meetings are all available online. Is that right? Not online yet, okay. But I think they could be if people were interested. And we covered a lot of ground, okay? We heard everything from, you know, mirror image DNA containing organisms to, you know, people advocating for plants or their favorite bacterium. We heard a lot of really cool projects about how you could engineer a genome by adding in a number of pathways and so on. But I didn't find adding pathways, those types of projects, they didn't fit the bill for me because they're not genome-wide and pervasive genome engineering. And so at some level, we came back to what we proposed in science paper, but with the, I think, added twist that recoding for virus resistance was a great theme that could have a really big practical impact, not just for human cells, but in animals and plants as well. What was the process like? Basically, I kept coming back to how important it was, I thought, for us, GP Write, to put such a project together, and encouraging people to think about it. And at one point, I kind of took matters into my own hands and said, okay, I've heard two projects that I think fit our bill and have merit. One was essentially the ultra-safe cell line with some added bells and whistles, and the other was to basically make the sort of archetypical human genome that would have the ancestral allele of everything. And I pitched both of those to the group, and there was overwhelming support for the former and not much support for the latter. And so that's pretty much how the decision-making happened.
Good morning. Two questions. First, have you identified the institutions that will be doing the work on this project? Are they all academic labs, or will there be some companies involved? And the second question is, how will this project be funded?
So I mentioned on my slide there's 106 institutions involved, or laboratories involved. It's a pretty good, healthy mixture of academic, non-university academic, and companies. We want this to be as open as possible. Recognize there will be some things where a service will be provided where we don't know exactly what's happening under the hood, and that's okay. But it also doesn't stop us from engineering that to some other method that's more open. So I think right now we have a healthy community. Who's paying for it is we are putting it together with available funds, like Jeff and I have described. National Institute of Health, NHGRI, SEGS grants, Center of Excellence Genomic Science. Those are two very it's a very forward-looking program that Jeff Slosh was involved in establishing. But there are many others too that have helped start synthetic biology as well as GP Write.
So just to follow up also, I think, probably are 106 of those 106 labs going to step up to the plate and say, I want in? Maybe not. Our experience with the yeast genome was, there we had 16 chromosomes to give out, and it was sort of a nice way to package pieces of the genome and yet still give a team ownership of that chromosome, which I think is really important. So we envision a similar sort of thing where, you know, probably the number of teams that is really going to be in a position to deliver and get their own funding, because that was part of the deal with the SA 2.0 was it wasn't enough to say, okay, I have a lab, I know how to do molecular biology, et cetera, but you had to be willing to go to your own government or however, your university, and get the funding in place to do your chromosome. So again, we haven't made any final decisions about implementation, but that would be one scenario for how we might proceed.
Could you remind us what the international distribution is of the teams? Or the participants?
You mean for GP, right? Oh, maybe Nancy can answer that.
15 countries. There is a slight bias towards the United States, as you might imagine.
And China.
Brazil just joined as we speak.
Joe?
Have you considered bioprocessing cell lines like CHO, where adventitious agents and things like that continue to be a big problem?
Anybody else? We did consider CHO, and it's still a possibility. What we were trying to do is get the technology to the place where we can do this again and again. We do human, we do CHO. You can ask, why is it that CHO is so special, and transfer all those special things into the new line, or make them even better. I think one of the things that people would really like to have is virus-free cells. If you look at what the FDA approves, they get very fussy when you use animal serum, even briefly, on your cell line, because of the examples in the past where they've had to deal with viral contamination. So if we had a virus-resistant cell line, that would probably answer some of the, but you also need productivity goals as well. We haven't written any specifications on productivity, and that would be great. So in a way, we're more interested in the specifications, because we're not going to get a free pass by the FDA to say, oh, these are CHO cells. They're going to say these are not CHO cells, right? If we've changed 230,000 positions. So we might as well put the specifications of what it is we like about CHO cells on the spec sheet right now. So we're welcome the community to do things like that.
I think underlying your point is, and I think we took this into account, is the potential commercialization around biologics, which is ever growing, and hopefully this will be a major resource for that as we go forward, not just recoding T cells, but making biologics, which is what George is really talking about. And so maybe importing those properties and making a better cell line might be an interesting way to go as well.
And I would just add, sort of to your point, as well as a few of the others, that a lot of these details in terms of cell lines and actual approaches and specifications to recoding, I think are still very much a work in progress. And I could easily see some very focused proof of concept efforts needed to really ask the question, should we go after these types of codons versus these other codons? What are some of the limitations of placing LOXP sites? How does that impact, splicing sites in human genes? So I could see a lot of very targeted proof of concept projects across different types of cell lines being launched in the near term to ultimately converge on a much clearer path going forward that would probably mirror something that Jeff described for SC 2.0.
Of course, an incredible success would be, oh, it turns out we can actually do it really well and really fast, and then you could do it on any cell line, and we wouldn't be having this discussion.
So in George's introductory talk, you mentioned the fact that we haven't sequenced any genome completely yet. So if you want to do this kind of a project on whether it's a PGP cell line or a mouse or wheat, how are you going to deal with the fact that we haven't completely sequenced any of these genomes? How's that going to get in the way?
We haven't sequenced any major animal or plant species. We have done some microbial species. I think that should be a side goal, definitely. It is certainly one of my obsessions to do that. But if I had to place money on it right this second, I would say probably some combination of long reads like nanopore and in situ sequencing, where you can trace, like the Wu lab here at Harvard has developed oligopaints where you can trace the chain from telomere to telomere. Some combination of those would probably allow us to finally get a diploid complete genome. Well, actually, I think the effort dropped off precipitously after 2004, and we did our final declaration of victory. That's true. Yeah, and they have finished one chromosome, which is the human Y chromosome. So it can be done. It has a fairly short centromere.
Y chromosome's always cheating, though.
Yeah. But anyway.
Same thing with deleting the Y chromosome. That's, it's interesting that they did that, but it turns out the Y chromosome is lost spontaneously at a very high rate. And I wouldn't be surprised if when you try to delete one copy of an autosome, you're going to hit some rough sledding.
I think they did 21, or a different group did 21. But anyway, I think it's a really excellent point you make. And part of it has to do with motivation. Yes, a few groups have been continuing it since the 2004 victory. But I think now we have a real reason to finish those regions, because it's very hard to edit or write without being able to read. If any of you've ever tried doing that, try writing a novel in Japanese if you're not a native speaker. So I think we need to finish it, and we need a 3D structure too, as Jeff nicely pointed out.
The good news, though, is that as far as we know, all the genes are, all of the protein coding regions are not within those regions. And actually, if we do the recoding for virus resistance, we don't really need to delete the transposons because they'll be inactivated at the same time. We're not going to recode the transposons, right?
Well, we might actually have to recode the envelope gene of the ERBs, the herbs, because those might have a function. We have specifically avoided touching those in the pig because they have a role in placental function. So we have to be careful about even the repetitive elements, that we might want to recode some of those. And we might want to change some of the repetitive elements for the purpose of stability or knocking out centromeres or something like that. So maybe not absolutely essential, but I hope this will motivate the broader community to finish up a few genomes.
Got another question over here.
So, two organizational thoughts. One is, since we're thinking about this project having, each group bringing its own funding, I hope that the leadership will organize it in such a way that there can be a rolling entry of groups, because some labs, I'm certain, are ready to go right this moment. Some may be in, a multi-year process to be able to get funding in place, and it would be good to make sure that people can enter as things go.
Yeah, I think one of the challenges we're going to face there, I think it's an excellent point, is that, and we saw this, and I'm totally guilty of this on the yeast project, was the people who start out grab the small chromosomes right up front, right? Because they can declare victory more quickly. Well, look how long it's taken us to finish chromosome 1, by the way. But anyway, I know that there are already multiple teams working on 21, 22, X, and Y all over the world, and that's kind of a big, giant waste of effort because, well, maybe not entirely, because they're probably doing different designs than we're talking about right now, and they might learn something really interesting. But from the perspective of the project, I think that's going to be a really kind of tough decision is, like if somebody's really claiming that they're ready, up and running to do this right now, I think they should take the biggest chromosome, not the smallest chromosome.
Well, the other thing I think we can do is help organize people to do joint funding, because there's probably going to be a lot of opportunity, you're probably going to allude to this, opportunities, at least in the US, for large-scale joint funding efforts, and oftentimes coordinating those is the hardest part.
Can you be more specific? Because I'd like to hear more about that.
In my dreams. I'm thinking.
The other thing, again, along these lines, that the second point I wanted to raise, there is an element in IP, and what are the rights that are going to be attached to products of this, especially because we're talking about crossing international boundaries. And on the one hand, there's a very strong scientific reason to want to keep things as open as possible, especially final products of this work. On the other hand, we have a lot of companies involved, and having too strong an openness may inhibit corporate participation.
We've seen a lot of corporate enthusiasm. Maybe it's because their goals aren't fixed in stone yet. Hopefully they won't be fixed in stone even at the finish line. But I think there's plenty of room for people to patent processes and maybe even intermediate parts, as has happened in many other private-public partnership projects. I think it will be mostly pre-competitive is the term that's used in the pharmaceutical industry, where everybody can benefit, and so there will be either patent pools or just make it open. I should mention the Personal Genome Project cells are open in the sense that nobody in the Personal Genome Project profits from them, but they are consented. The individuals that sign up are consented for their cells to be used commercially freely. In other words, anybody, it's like Wikipedia. You can make a commercial version of Wikipedia, but Wikipedia doesn't manage that.
So we obviously addressed this issue with the SC2 project, and I'm sure some of you know about this, but it was a requirement, and we could talk about doing the same thing with GP Write. It was a requirement that what we defined as primary IP, which is the sequence of the designed genome and the yeast strains that contain those sequences, would not be patented and would be always open for free distribution to qualified parties, but including companies, okay? But then secondary IP that was developed by the teams in terms of how to build it, how to use it, was completely available, and that incentivized the teams to participate. And every team had to sign onto that, including our corporate partners. And some of the teams didn't want to do it because they said, well, what about this primary IP stuff? I'm not signing up for that. And so we just said, well, take it or leave it. And it's worked out really well.
Sounds like a really good start.
I think we have time for one more question. So regarding the testing part, which I think is one of the biggest challenge for GP Write, how do you think we can design a cell line or a genome such that is a testing platform that is useful for as many applications as possible? Because one of the interesting things, it would be, okay, I have a cell line where I can knock in any haplotype of my cancer GWAS and see what's going on. But is it going to be a platform that give me useful information, or is it just going to be some sort of artifact like when we test in cell line?
I think one of our criteria for picking projects, and again, this is not carved in stone, was that it both be intellectually interesting and practically important as well. And I think human stem cells have the potential of being able to both either directly manufacture or be turned into a different kind of cell that can manufacture pharmaceuticals, vaccines, and so forth. And they might also be useful for transplantation and for testing of hypotheses, testing therapies. So that's a lot of uses. It may not be all the uses that you have in mind, but we are really open to suggestions about how to up front make the choice of cells maximally useful for academics and industry.